Am I audible?
Yes, madam.
Good morning to all. I, Dr. Dipti Dhere, working as Assistant Professor, Department of Microbiology, Dayanand Science College, Latur, and working as University Coordinator of Microbiologist Society, India, Swami Ramananda Tirtha Marathwada University, Nanded, welcome you all for the 35th lecture of PhD webinar series organized by Microbiologist Society, India. Microbiologist Society has created this virtual platform for sharing the research ideas so that microbiologists can work together for development of microbiology subject and its research aspect. Today, Dr. Shivaji Wagmare sir is our guest. He is working as assistant professor at Department of Microbiology, Ferguson College, Pune. He is also working as nodal officer, academic bank credit. He has completed his PhD from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. He is also a PhD guide. He was awarded with Golden Jubilee University Junior Research Fellowship for doctoral study. He also achieved a first prize for poster presentation in Avishkar 2010 at Zonal Research Competition. He is life member of Microbiology Society India, Association of Microbiologists India, and life member of Indian Science Congress. He has total 8 research publications on his account. He has published his research in 7 national and international conferences. He also served as resource person at various national and state level conferences. He has completed his research orientation and faculty development programs and also organized conferences, seminars, and workshops. He is working as coordinator of many committees at college and university level. With this very brief, I request Dr. Shivaji Wagmare sir to present his research. Sir, you will be provided 45 minutes for your presentation, and then we will have the 10 minutes question answer session. Over to you, sir.
Okay, thank you, madam. Thank you for your brief introduction. Good morning, friends. Today, we are going to learn or focus on a very interesting topic. I performed this work in 2010 during my PhD work under the guidance of respected Dr. M. Deshmukh sir. This work is completed in Aurangabad, subcampus Usmanabad, the Dr. Babasaheb Ambedkar Marathwada University. The lab, whatever the construction was going on at that time. So I was the first student at the department who did my PhD in Usmanabad subcampus. Dealing to this topic, biosynthesis of metal nanoparticle, there are the many questions arise or the why we select this topic for our research. Right now, we can see the very much research is going on nanotechnology or on bio-nanotechnology. So dealing with this subject, we need to construct the various devices. Those devices are going to work in a very small space, but they can give a very functionalized applications. We are going to use them in a medical field, or it can be used in the various environmental aspects. Keeping all this view in mind, I have selected this topic for my research work. The journey of this nanotechnology field is started from the very famous lecture or the famous talk by Sir Richard Feynman. In one very famous lecture, Richard Feynman said that there is a plenty room at the bottom. The meaning is that the ones we are going to be constructing any material, or once we are going to be observe any material, rather than its bulk form, once we are going to observe it at a nano form, it has various properties. With those properties which are not going to be seen at the elemental form or its molecular form. So these properties are going to be seen is there at atomic level or the atomic form. And these properties, this functionality are going to be used here in a future for various aspects. Generally, the nanoparticles are going to be defining those particles which are the dimensions or the size in between 1 to 100 nanometer. Generally, we can see the synthesis of nanoparticle. What is the reason behind the synthesis of nanoparticle? Because these particles are too smaller. They are there in the nanometer size. Hello. These particles are.
Hello, Nupur Goel, please mute your mic. Nupur Goel.
Shall I continue, sir? Deshmukh, sir? Okay. So the synthesis of nanoparticle, there are the several reasons. Because of this, we are going to be moving towards this field. These nanoparticles, or these particles, are the faster, lighter, cheaper, and they can show the more functionality in a small space in a large area.
You can proceed.
Yes, thank you, sir. The properties of this nanoparticle changes with the change in the size of the particle. So it may be some properties like the color of the various compound, conductivity, boiling point, melting point. These are the properties that we have been studied. It might be in a chemistry, and these properties are going to be used for the applications of a various material, or it may be the crystal shape, magnetism, chemical reactions are going to be changed once comparing with a bulk form to its nano form. You can see this is one example for this. The compound which is there in all these 5 tubes is the same. Only change in the size of a material, you can see the change in the color of the material. So this is what you are going to be seeing here. This is the gold, but change in the size of the gold particle, it can show you the change in the color of the particle. Nanotechnology involves now here the tailoring of material. That means we are going to be constructing this material at an atomic level to attain these unique properties. It can be used for the various other purposes like the microelectronics, optical properties, catalysis, drug delivery, and it can be used for the many other purposes in a medical microbiology. Now, this is a brief introduction regarding the nanotechnology. Now we are moving towards the actual synthesis of nanoparticle. In the synthesis of nanoparticle, there are main three methods. One is the physical, chemical, and the biological method. So once we use the physical method or the chemical method, there are the several drawbacks. Like we are not going to be concentrate on a particular size and the shape of the nanoparticle. But here in a biological method, we can concentrate, we can fix up the size, shape of the particle. That's why the scientists who are working in the field of nanotechnology, they are moving towards the biological field for the synthesis of nanoparticles. In the biological synthesis, there are the several aspects. So we can use the live cell biomass. We can use the live cell biomass, or we can use the dead cell biomass, or we can use the product of a microbial cell or the product of other organisms. It can be used for the synthesis purpose. For example, DNA, protein, viruses, these are the viral particle. They can also be used for the synthesis of nanoparticle. So as I said, there are the several disadvantages or some drawbacks while using a physical and a chemical method. Along with this, these methods are the hazardous. They are not eco-friendly. That's why the eco-friendly method, like a biological method, is going to be used for the synthesis of nanoparticle. In biological synthesis, wide variety of different organisms. It may be the plant, it may be the fungus, it may be the yeast, bacteria, algae, even human cells, it can be used in the synthesis. So within this, the there are the very dominant species or the dominant genera, microorganisms, which are there here in the ecosystem. These are the actinomycetes, which can be used for a variety of different purposes. Because actinomycetes is a very popular organism. It can be used for to give the solution to the nature in concern with the to solve the problems of the waste management or to solve the any issues which are there here in a ecosystem. So that's why I have been concentrated, I have been selected the organism is the actinomycetes. Now this actinomycetes is the group of a bacteria. This is a gram-positive organism, and this bacteria is going to be used for this purpose. Previously, this organism is classified under the group of fungi because of their mycelial structure. But later on, now this is going to be classified under the group of bacteria because of its content.
Right.
Several work is going on this particular aspect.
Chai patti.
The actinomycetes. In considering all these things, now aims and the objective of my study is to isolate different actinomycetes, from the eastern Balaghat ranges of Maharashtra. So we are going to use this actinomycetes where this eastern Balaghat ranges of Maharashtra is situated here.
Requested to mute your mic.
Ma'am, can mute all so that then Wagmare sir can unmute himself. Yeah. Co-host can do that.
Just a minute, madam, I'll check.
अर्धा किलो
Sir, now you can start.
Hello.
Yes, sir, you can start now.
Okay. So the keeping all this view in mind, we can use this actinomycetes for the several purposes, like to give the solution for the degradation of the various dyes, which can be used for the biostimulation, bioaugmentation activities here in the soil. So I have been used this actinomycetes for the synthesis of nanoparticle. For that, I have been screened out as organism from the eastern Balaghat ranges of Maharashtra. I have been checked their metal tolerance to the metals, example, gold, silver, manganese, magnesium. So these are the metals I have been used for my study. And later on, once the tolerance of this metal have been studied, these organisms have been used for the synthesis of nanoparticle. After the synthesis of nanoparticle, these materials have been used in the characterization by using UV visible spectrophotometer, XRD, EDX, SEM, and the TEM. So these are the tools and the techniques have been used for the characterization of nanoparticle. So there are total 40 actinomycetes were isolated from the soil, studied for the metal tolerance, 4 selected and identified up to the species level to study the metal tolerance ability. So I've been checked here in a medium where without any sole source of a carbon, I just given the metal solution to the organism. And some of the organisms are there, even though some actinomycetes are there, these actinomycetes can grow in distilled water. So along with distilled water, I have been added the metal concentration, and you can see the growth pattern of this organism have been seen. So those organisms which can be shown the high growth, these organisms are going to be used for the further study. So likewise, I have been used or carry on the study for the metal like a silver, metal like manganese, zinc sulfate. For this purpose, I have been used. So these are the plates of the 4 cultures have been used for the study. You can see this actinomycete colony. They are the very tough colony, very hard colony that you are seeing concentric growth, concentrated towards the center. They are the sporulating organism, and these organisms have been used for my study. While performing morphological study along with the biochemical characteristics, so I have been use this organism for the SEM. So in SEM, you can see the very beautiful arrangement of the spore along with its mycelia. So you can see these are the mycelia, and this is a spore arrangement. So this is the arrangement of the spores. This is the identification criteria. So it may be the whether from the which genus this particular species is. So just like the 7 different genera are there in the group of actinomycetes. So from that, this is the streptomyces. You can see this is spiral form of the spores. It can be seen here. So along with this, it may be some spikes that can also be seen on spores. So these are the identification criteria for the actinomycetes. These are the results for the biochemical characteristics. So just like a bacteria, because actinomycetes itself is a bacteria, we can proceed for the biochemical characteristics. So it may be the sugar fermentation, sugar utilization, enzymatic test. It can also be carried out for the identification of these organisms. Along with this, genetic pattern or the genome analysis is most important. And then only we are going to be conclude which species is. So at that time, in the 2010, we have been given the culture to the NCBI to analyze the DNA of this metal tolerant organism. And so that time we have been seeing the matching of this organism, how much 99% of matching of this organism, and accordingly we are going to be conclude the which species it might be. But up till now, this organism is not going to be identified up to the species level. But you can see the location of this organism is there here in between the Streptomyces species. 2 Streptomyces species are there. So this is the K5 is the code number or the code of my isolate. So likewise, I have been identified these 4 organisms by using its 16S rDNA sequencing pattern. This is K27. So you can see the location where this organism is present. Likewise, the identification has been done, and along with this genetic pattern, I've been used some biochemical test and then confirm that this organism it might be belonging from this particular species, and these organisms have been used for the further study. We optimize this organism because for the study I need to require a more biomass, mass production of the biomass, because here in biology or here in the microbiology you are using a cell biomass for your study. And to need the cell biomass, you need to produce or you need to grow this organism so that this organism can give the huge biomass. For that purpose, you have to use a production medium. So within which you will get the luxuriant amount of biomass. And likewise, I've been used it for the various pH and the various temperature. So I found that in a pH 7 and the temperature is near about the 35 degrees Celsius temperature, this organism will give you a very good result, very good biomass. And this biomass have been used for the further study. So for this purpose, this cell biomass, you need to clean out by using a double distilled water. You are working with the metal solution. So you need to clean up the biomass properly, wash the biomass properly so that those nutrients which are adhered to the microbial cell or the mycelium. So these will get washed properly. So for this purpose, you need to use double distilled water. If there is a problem in your distilled water, it may create the problem of a contamination of a metals, and you will get the false results. To avoid this, better way to use a very good quality of a double distilled water. And then I have been exposed this biomass to the metal solution. This metal solution is going to be prepared in a various concentration, starting with the 0.1 molar to it may be up to the 1 molar in various concentration. I have been just exposed this biomass to the metal solution. So within a 24-hour or up to the 48-hour, there is a change in the color of the biomass. So this change in the color of biomass have been used for the further study in various research papers, saying that the change in the color of the biomass from the previous one. So whatever your previous color is, so previous color of the biomass, suppose is the dirty white color or the pale white color. So this white color, it may get changes it into the faint pink, purple or the violet. You can see after 12 hours, 24 hours, 48 hours and the 72 hours. So there is a change in the color of biomass. So that indicates there is a change in the size of a particle. So this is the first step or the first report, first evidence that the nanoparticles are going to be synthesized in your sample. Now, while using this particular data, you are not going to be confirm the what will be the size of the nanoparticle. So for that purpose, you have to carry out the further study. So it may be the UV visible spectroscopy. You have to carry out the XRD, EDX, SEM, and the TEM, transmission electron microscopy. So with this study, you will get the information about the what is the morphology of the particle, what is the size of the particle, what is the polymorphic or the monomorphic nature of the particle. So this information you will get it through this study. So likewise, these are the biomass or these are the various organisms I have used for the synthesis of various different metals. And these are the change in the color that, so this is a preliminary step I have been performed there. Now here you can see this is a biosynthesis of gold nanoparticle. This is the previous color of the biomass, washed cell biomass of actinomycetes, where you can see the pale yellow color, which will get turned into violet in color. So now this is the without metal solution that you are going to see. So you can keep the control along with the metal solution, but don't incubate this. You can keep it in the refrigerator, and one you can keep it in incubator. So you can observe the change in the color of biomass. Those tube which we kept in the kept as in control in the refrigerator, you are not going to be seeing the color change. But those who kept here in the incubator, you can see the change in the color. So this is the evidence that the nanoparticles are synthesized in your tube. Same way here, I will perform the experiment for the silver nanoparticle. Now, the gold and the silver, these are the metals which can give you or give us the visible change, but not the other metals like the Mn or the Zn. These are not going to be seen the more visible color. So for this purpose, you need to go for the other methods or the other tools for the analysis like the XRD or the EDX. So this is the pale yellow color of a washed cell biomass, and this is after incubation of the silver nanoparticles. Same way here you can see these are the two different cultures I have been used for the study. Again, another example of the K27, you can see the change in the color. In the earlier, again, the dark brown, here the faint brown color which can be seen. It's a depend upon the type of organism. Only because the which type of the enzyme or the concentration of enzyme these organisms possess. Because in the synthesis of nanoparticle, there is a role of oxidoreductase enzyme, or this is a class of the enzyme. Oxidoreductase is a class of enzyme. So within which the which organism or the which enzyme is there is present in that particular organism. Accordingly, they are going to be show us the color sets. Now here this is the manganese nanoparticle. You can see here this is the control. So various concentration of manganese nanoparticle manganese, and I have been used the biomass, what cell biomass within this, and I can see the concentration of a metal. It can also matter because we know these are the heavy metals, and the heavy metals have a oligodynamic effect on a microorganism, so they can kill the microorganism if they are then the more concentration. So or if they are then the less concentration, they are the beneficial for organism itself. But they are there in the more concentration, they will be toxic to the microorganism. So make sure that whatever the concentration that you have been used, it should not be a excess or should not be toxic to the microorganism. Like the same, I prepared this for the zinc nanoparticle. UV visible spectra, it can clearly reveal, it can clearly show the peak at a particular position. So this peak at a 550 nanometer, this indicate these are the gold nanoparticle. So this is again the second evidence that can give us the whether the bulk compound will get converted it into the nanoform or not. So the first evidence is your visual observation. Second evidence is the UV spectroscopy. So within which, if you observe the peak at a particular position, so if it's there at for the gold, it's a 550 nanometer. So likewise, here you can see the peak at the 450 nanometer for the silver. So that is the very good evidence that the nanoparticles are synthesized in your particle. Like the physical method or the chemical method, you can see the very sharp peak. But here in the biological sample, you not get the sharp peak because in biological sample, there are the other entities are there. These entities, it may get interfere. So that's why you're not going to be seeing a very sharp peak like a chemical synthesis of a nanoparticle. But these are the concrete evidence that the nanoparticles are synthesized here in your sample. Likewise, the manganese and the zinc nanoparticles can also be seen the peak at the 380 nanometer. Here also you can see the peak at near about the 400 nanometer. So these are the evidences that the nanoparticles are synthesized in a sample. Along with this, the next aim or the next aim of the study or the objective of the study is the to check the or to optimize the at what concentration or what pH will get the more synthesis of nanoparticle, right? So like for that purpose, I will use the various different concentration, various different pH, temperature, and we can see the synthesis of nanoparticle once we are going to be revealed with a SEM study. So you are going to be seeing here the along with the mycelial structure, you can see some deposits. So these deposits of the nanoparticles are adhere to this. Partial for this again, I went for the partial purification of a gold nanoparticle. So for this purpose, I have been dried this synthesized nanoparticle synthesized biomass. So this biomass will be dried, and again I will use it for the furnace, and I just burn the whole content up to the dry. So whatever the remaining is the ash, this ash containing the nanoparticle. So this ash is nothing but is a purified form of a nanoparticles. So this is what you are going to be seeing at a SEM study. After the purification of this biomass or the nanoparticle which are there in the biomass, we are going to be seeing the uniformity in a size of a particles. Later on, the we need to go for the next third evidence, whether the nanoparticles are there in the sample, and if they are there, what will be the size of this particle. So for that purpose, the tool we can use is the X-ray diffraction pattern. So while performing with X-ray diffraction pattern, you can see the sharp peak along with the position of this peak. It can indicate the size of a particle which are present in your sample. So these are the monodispersed particles are going to be seen here. Monodispersion and monomorphic particles are going to be seen. Most of the particles are the in a unique size or uniform size. So here, for that purpose, to know the size of the particle, we can use the Debye-Scherrer equation. So you will get the exact size of the particle. Again, the fourth evidence to check the gold nanoparticle or the silver nanoparticle which are there in your sample. For that purpose, EDX is going to be prepared. Now here, this peak indicate there is a position of the silver, and where it can give us along with this silver, you can see that there are the other peaks. So these peaks are of the other elements like carbon, oxygen, sulfur, phosphate, and other metals are also seen here, because we have been used the biomass of the organism. This is organic matter, because biomass it is composed of a carbon, oxygen, sulfur, potassium. So these are there here in a biomass. So that's why you are going to be seeing here the other signals. So once we nullify these signals, you will get only the peaks of the metal that you have been used for the synthesis of nanoparticle. Likewise, the same study is going to be carried out for the gold nanoparticle, and very beautiful structure that you have seen under the transmission electron microscope. So you can see this is what the before the exposure to the nanoparticle, before the exposure to the metal solution, what you are going to be seeing here. So this is the bulk form of the metal, and this is what you can see after the synthesis of nanoparticle. Here, the particles are going to be bulk form is going to be converted into the smaller part. So where this bar indicate, this is the size bar, this is 100 nanometer. So where you can see the whatever the particles which are synthesized are less than 100 nanometer, and this is what our aim to convert the bulk form into the nano form. So this is going to be used for the other cultures or the other isolates. Here you can see the conversion. Now, this organism or these particles, even actinomycetes are going to be shown the antimicrobial effect. So here our aim is to check whether this gold, silver, zinc, or the other metals which can show the antimicrobial effect or not. So for that purpose, we have been used some of the test organisms for this purpose. So these test organisms are going to be obtained from the NCIM, and these organisms are going to be used for the study. So where we can observe whatever the form of the particle. So it may be they are there in the bulk form or in the nano form. So how they are going to be affect on a microorganism. So here you can see this A stands for this is the AgNO3 as a control, and we know the AgNO3 silver nitrate has its own antimicrobial spectrum. It can be and act as an antimicrobial. So in the B you can see actinomycetes. So that means the silver biomass with the silver nitrate. So at 0 hours we are going to use it for this purpose. So you can see here this is biomass as a control we have been used, and then the T is going to be used. This is the AgNO3 treated biomass after 72 hours without sonication. One is there without sonication, that is a T, and S is there with sonication. So you can see if the nanoparticles are synthesized, nanoparticles are not synthesized, only the effect of biomass is there. So first one is the effect of biomass, second one is the biomass along with the nanoparticle, and third one is the biomass which is a sonicated, because once the nanoparticles which are there inside the biomass, which will get come out from the cell, and which can show us the antimicrobial effect. So we can see, so for the three conditions, for only for the silver, it must show the some antimicrobial spectrum. Along with the biomass plus the nanoparticle, it can show the some effect. And the third one is the sonicated form of the nanoparticle. So we found that those which are there with the sonicated form, so these microorganisms which will burst open, the cell burst open, and then the nanoparticles will come out. So that's why you are going to see the more antimicrobial spectrum, or this diameter that you are seeing here for the sonicated biomass. So this is what the graphical representation of these results that are going to be seen here. So in this, we can conclude that definitely the rather than the bulk form of a compound or the material, this nano form of a material, they are the more effective. So in future, we are going to use it for the other purposes. So concluding this, in summary, actinomycetes have ability to produce a metal nanoparticle when the biomass was treated with a metal solution. Visible observation was carried out for the desirable color change after the exposure of 72 hours. XRD pattern, EDX, and the SEM, these are the concrete proof that the particles are synthesized, and they are going to found is near about a 20 nanometer to, it may be the wide variations are there. So the 20 to 25 nanometers size of the diameter, they are going to be produced there. And these organisms are going to be shown as the antimicrobial spectrum. Now the scope of this study, so it can be, we are going to be using this study for to find out the exact mechanism of synthesis of nanoparticles. Right now, we know that various enzymes, so those enzymes are going to be there. There is oxidative reductase class of the enzyme, along with the other peptides. They can also be involved in a synthesis of nanoparticle. So clear-cut mechanism by which these microorganism and the product of organism, they are going to be converting the metal into the very a smaller form, or the nano form, that we are going to study there. To study the mechanism of synthesis of nanoparticle, then the biosynthesized materials with the desired size and the shape. So for this, there is a lot of scope in this particular study. Assembly of the particles and the nanomaterial. Study the silver nanoparticles as an antiseptic paste as a therapeutic agent. So right now, it might be in the market, such a type of products are there. Nanogel is available here in the market. Along with this, we can check whether any role of the any pigment in the synthesizing the nanoparticle, like if they are there in the protein or they are the peptides. So what would be the role is there in the synthesizing the nanoparticle? And the melanin as a nanoparticle, it can also be studied there. So this is the scope of the study. These are some references have been added here. Thank you. If any question or any discussion, this is open for the discussion.
Thank you, sir. It was very nice presentation. And the session is now open for question and answers.
There is one question in the chat box. How does bacteria react with the metal added? What is the mechanism between the bacteria? Just now I told it may be just like we know the ETC, electron transport chain. One salting in, salting out. So there is some porine proteins are there. So they can give some entry of a microorganism. They can give the entry of some metals, because that is required to stabilize the cell. So it may be from that way, or from that mechanism, there might be some entry of these metals within a bacterial cell, and they are going to form an intracellular synthesis of nanoparticle, because it can be formed either in intracellular or in an extracellular way. So it might be that some of the proteins which are there in an intracellular way, they can be worked there. How you measure the OD on a UV visible, it is a filamentous organism. See, after having used that, just by filtering it, after purification, I have been used the wood content and I have been checked there whether they are going to be shown any observation there or not. Is it bioaccumulation, bioadsorption, or the biosorption? So, see, as I said that there might be various mechanism because the exact mechanism is not yet clear. So, whether it's a bioaccumulation, bioadsorption, or the biosorption is there, but definitely it can be a bioaccumulation, it can be bioadsorption, biosorption. It depends upon the type of the enzyme and the other peptides they are going to be used for the synthesis purpose. How to recover the synthesized nanoparticle? This synthesized nanoparticle, it can be recovered just like the formation of ash, as I mentioned here. So you can get it in a dried form of a biomass after the synthesis, and you can convert this dried form of a nanoparticle. You can just burn it, and you will get the nanoparticles. What concentration used for the silver gold nanoparticle? What is the lambda max? See, the concentration is going to be used for this purpose. You can go it 0.1 molar, or you can use it to 1 molar. As per the tolerance limit of your organism, you are going to be use it for this purpose. Lambda max, again, you are going to study it on the basis of is a trial and error experiment. So you will get the lambda max.
Any more questions?
Does the nanoparticle have any health effect? Yes, definitely. You are asking it for the positive effect or the negative effect, but it can show the both effect, either the positive or it can show the negative effect also, because some of the studies are going to be claiming that the nanoparticle, it can cross the placenta, and some you can see they are the very effective, just like they are going to be used for the nano drug delivery. Nanoparticle is a cost effective. Yes, slow nanoparticle, they are the cost effective because they can give us the more impact or the more good results. Can cyanobacteria be used? Yes, cyanobacteria can be used for this purpose. As I mentioned, there's some of the representative organisms. Some examples have been used, mentioned there, but you can use the variety of different microorganisms for this purpose. So cyanobacteria can also be used.
I think there are no more questions. Shall I proceed to next session? Next part of this session, sir?
Yes,
yes, you can proceed.
Now it is time to express gratitude. I express my gratitude to Almighty God for giving me one more beautiful day to contribute myself. I am thankful to Professor A.M. Deshmukh, President, Microbiology Society, India, for creating this virtual platform for all of us. I am also thankful to Professor A.M. Deshmukh, sir, for providing me opportunity to host this program. I am thankful to Dr. Shivaji Wagmare, sir, for presenting his research work in very simple and lucid way. Thank you, sir, for a very nice presentation. I am also thankful to all participants of webinar series. And with the permission of chairman of this program and guest, I declare that this session is over.
Thank
Thank you,
you,
madam.
Dipti.
Thank you, sir.
Thank you, sir.
Yes, you have done a very good job today.
Thank you, sir.
Thank you. Thank you all for attending and interacting with the speaker.
Thank you, sir.
Yes, and thank Dr. Shivaji for a nice presentation. And you have inspired many young workers in microbiology. Thank you.
Thank you, sir. Thank you.